Hi, my name's Rachel. I am a third-year PhD student. I'm part of the Human Toxicology program here at the University of Iowa, and I work in Dr. Ethan Anderson's lab, which is part of the College of Pharmacy here. And this is what a typical day looks like for me. I usually start my mornings off with a pretty quick breakfast and getting a lunch ready to go. To get onto campus in the mornings, I usually take the bus. My apartment is right on a bus route, so it is a pretty quick walk to the bus stop. I am just walking up to the College of Pharmacy building. As you can see, it's about 8:30 in the morning now. This is pretty typical time for me to get here though. This is the inside of the building as you walk in. This is actually one of the newest buildings on campus right now, so everything is really nice. As you can see, we're passing some classrooms, a PharmD lab. Over there is our big lecture hall, and there's plenty of room to sit and just work, which is really great too. My lab is up on the 5th floor of this building, and this floor is fully reserved for research and is where I spend most of my time. On this floor, we have this conference room, which is often used for lab meetings. We also have a lot of office space for both research students and for faculty here. I am scanning in to get into the office space reserved for my lab. As you can see, it's a pretty nice space in here, and each student is given their own desk and computer to work with. This is the lab space where we do all of the research. This section is split between two labs. So this back half is the Anderson lab area. All of the instruments in our area are typically just used by the Anderson lab, but all of the labs in this building usually don't mind sharing amongst each other. Each of us are also given our own lab bench area as a space for us to do our work, and so this is my area. Today I am doing a longer experiment where I'm going to measure mitochondrial respiration and membrane potential. This will take up pretty much my entire workday. As a third year though, I am not taking any classes this semester, so it does free up a lot of my time to be able to do these kind of experiments, and I am currently preparing some buffers that will be used in the mitochondrial isolation part of today. I really enjoy the research I get to do in the Anderson lab. There is a big emphasis on metabolism and biochemistry concepts involved in our lab's research, and that really is what interested me in the lab. I have finished isolating the mitochondria and am preparing an absorbance assay to determine the amount of protein in my isolations. While that is finishing up, I will take a quick lunch break in the break room that's found on our floor. As you can see, the views from this room are a big plus. I'm now beginning my assay where I am measuring respiration and membrane potential in the mitochondria that I isolated earlier. I decided to get my PhD because I really enjoyed the research I did as an undergraduate, and I wanted to continue in that line of work. The financial support that's provided to PhD students was also a big plus when considering this degree. My tuition and a stipend are supported by the Anderson Lab and by any grants that I've been awarded. I'm currently on an NIH training grant. I'm finished up for the day. It's about 5 p.m. and I'm walking home. It's only about a 20-minute walk to my apartment, so that's pretty nice. I have a rec league volleyball game tonight in Crawford, which I'm going to go get ready for, and then after that, I'm pretty much done for the day.